Hi Abhijit, good morning. Hi Moksina, good morning. Good morning everyone. Hi Srujan, good morning. Kusum, good morning. Sorry to make you all wait. Good morning, good morning. How are you doing all? How are you? Fine. How's your mental health? Is it calm, cool, pressurized? कैसे हो आप लोग? Okay, good morning. Hope you all have your had your breakfast. मेरा तो नहीं हुआ है, एटलीस्ट आप लोगों का तो हुआ होगा. अच्छा, ठीक है. थैंक यू. थैंक यू. Okay. So Srijan specifically wanted mutation along with this population and evolutionary genetics. So because of him, I also included that part in this population and evolutionary genetics. And plus, few students wanted numericals. I've just included one like question of the from the numericals part, but I would seriously suggest you like there are only like 2 to 3 videos that I've uploaded in the previous year, but the videos that have been uploaded in the 3 parts, the numericals, so they cover almost like every type of question that is asked in the genetics and plant breeding. Okay, I have almost covered each and every type of question that is asked in the genetics and plant breeding. So it will hardly take you like 1 to 2 hours to cover that part. So if you are specifically not strong at numericals, if you are specifically not confident about numericals, I would suggest to watch that numerical series without fail. The link will be available in the description box once the live stream is ended. Okay, so please make sure to watch it once. If everything is done, your revision is done. Even though like you are stuck with your revision, not able to complete much of the things, still go and watch the numerical video. That will definitely help you. 1 or 2 questions might come from the numericals part. Okay, and whatever comes, it will be definitely easy, so you can easily cover it. Okay, start करें. Can we start? Okay, fine. So population and evolutionary genetics. I'll try to make it as simple as much as possible. I'll try to make it very simplified, and we are not going to discuss anything in detail. Whatever is required, whatever might come in the exam, so we'll try to focus on that. Any marathon for the plant science. So this is the marathon of plant science. So what's your doubt? Okay. So population genetics. So let's start with the topic of population genetics. So what exactly, like before directly jumping into the population genetics, you have to understand this thing. See, the term is like biometrical genetics. See, what is biometrical genetics? It's a study under genetics which uses the mathematics or the statistical concepts or the procedures to study the inheritance. So this is what exactly the biometrical genetics means. Okay, which is combined with 2 different words, which one is like biometrics plus genetics. Genetics, you already know, it is like study of heredity and the variation. But to study the heredity and the variation from generation to generation, if we are using the mathematics or the statistical concepts and the procedures, so that becomes our biometrical genetics. Now it is broadly classified into 2 different fields. One is population genetics, other one is quantitative genetics. So quantitative genetics, I hope like you know like what is quantitative genetics, right? So population genetics, here this is a study of population. This is a study under genetics which mainly focus on what type of traits. It mainly focus on qualitative traits, opposed to what is quantitative genetics. Here it is like about the qualitative traits or the discrete variation for the trait showing the discrete variation. So this is a study of qualitative traits. How we are studying in the population genetics? We are using the frequencies of two important components. One is gene frequency and the genotypic frequency. How it is changing from generation to generation by the use of other words the allele frequency and the genotypic frequency. We are studying the population. So this is what exactly the population genetics means. Next thing, the quantitative genetics. Here the study is about the which traits. As I told you, like it is about quantitative traits, not the qualitative, which shows the continuous variation. But how we are studying, we can't study just by the allelic population, sorry, allelic frequency and the genotypic frequency. So since the genes are multiple and it is producing the continuous variation, we employ the statistical methods like mean or the variance mainly. So this is what quantitative genetics means. Okay, but remember, mostly like you will be seeing the term biometrical genetics that is used in case of quantitative genetics directly. Okay, but this classification is not given everywhere, but this is for your kind of understanding like how this is how it is, how this is it. Okay, how it is this, fine. So this is about population genetics and the quantitative genetics. Now in that we are focused mainly on the population genetics now. So once you come into the MSc, you will be mostly studying the quantitative genetics, which is like normally called as directly called as biometrical genetics. We do not use the term, we like you usually use the terms biometrical genetics for study of the quantitative genetics. So do not get confused with the terms. So now the population genetics. Population genetics, the study revolves under the two important factors I told you, which is like allele frequency or gene frequency and the genotypic frequency. Remember the allelic frequency or the allele frequency is same as gene frequency. Okay, there is no difference between these two terms, both are same. Do not get confused with these terms. Okay, I'll exactly tell you like what is the meaning of it. So why, first of all, what is allelic or gene frequency? It is a proportion of a specific allele within the population. That means like if there are total of 100 alleles, like what proportion is capital A and what proportion is small a, that is what the allelic frequency means. And what is genotypic frequency? Same, the proportion of specific genotype, like the proportion of specific genotypes like capital A, capital A, capital A, small a, small a within the population, it is called as genotypic frequency. So why it is important to study or to know the gene and the genotypic frequency? Why it is important? First, mainly, see, from the generation to generation, if we know about the allelic frequency and the genotypic frequency, we will be able to study the genetic variation. So for example, what I mean to say is, if I say capital A is 1, then what will be the value of small a? The frequency of capital A is 1. Then what will be the frequency of small a quickly? In a population, the frequency of capital A is 1. We are only talking about a single gene with capital A and small a as a allele. So what will be the frequency of small a? If you know the concept, like the frequency of capital A plus the frequency of capital A small a plus the frequency of small a, that will sum to 1. Okay, so this is the conceptual understanding because we are talking in terms of frequency here. Okay, now this means it is 0. So now just by seeing the values of this 1 and 0, can you tell me that there is a genetic variation in the population or not? Whether there is a yes or no, tell me, like whether there is a genetic variation in the within the population. Do we see any kind of genetic variation here? No. So that's why, that's how it is useful to know, to study the genetic variation based on the frequency of allele and the frequency of the alleles and the genotypic frequency, and to know the genetic makeup of the population. Just by knowing about the genotypic frequency, we will be able to know like what proportion of alleles are homozygous, dominant homozygous, what proportion of alleles are recessive homozygous, and what proportion of the alleles are heterozygous. Okay, and to know the genetic diversity. Similarly, same thing, like just like variation, if we are able to know about like what is the value of capital A, capital A, capital A, small a, small a, small a, how much diverse the population is, that we will be able to understand. Next, we can track the changes from generation to generation, one generation to next generation. So if you are able to track the value of capital F of AA or F of A from generation to generation, then we will be able to also track the mechanisms like natural selection, how something is favoring, how something is not favoring. Like for example, from generation to generation, the way like allelic frequency is changing, the genotypic frequency is changing, we will be able to know, understand the phenomena called like natural selection, the how the selection is processing, like if there is any mutation that is occurring or the genetic drift. All these evolutionary mechanisms could be understood by tracking these values. And for the study of the genetic basis of the disease, and we have like several disorders which are like a recessive homozygous, which are recessive in nature. Just by understanding the frequency of them, we'll be able to study the genetic basis of it. Whether it is genetically recessive or dominant will be known based on the frequencies of the alleles. Okay, that's the whole importance of the allelic frequency and the genotypic frequency. The next thing, very important thing to understand is population. So how do you define the population? Remember, population is always something we are talking about, the group of individual which belongs to the same species. The fundamental criteria is it should be belonging to the same species, but all individuals which are belonging to the same species all over the earth, that is it belong to the, is it called as a population, single population? So whenever we use the term population, it is yes the group of individual of same species, but where it is specifically inhabiting in a specified geographical area. That's the important thing. And the large population which is under the specific area again consists of small groups. If this is the entire area which consists of the individuals of same population, again these are fragmented into the different local population which is called as deems. Okay, and the deems, what is deems? Deems are nothing but the species made up of several Mendelian population, like which are fragmented with each other or which are isolated from each other. And this is the basis for the race formation. Yes, you know, right? Like even within the species of humans, like we have a different race. Like how it was done, it is based on the geographical isolation where they have resided in the different parts of the world. So based on which like the race formation has taken place. So this is what exactly the population means. Again, within the population, we commonly use see the term called as natural population in case of study of this genetics, population genetics. So what exactly is the natural population? What do we understand from this? Natural populations are the group of individuals which freely intermate, there is no restriction of intermating or there is no selective mating, and all the gametes of male have the equal chance to fuse with the female gametes. That's the most important one, the most important characteristic of the natural population which can be asked in the exam, which can freely intermate, plus every individual male gamete has a equal frequency of equal probability to mate, to fuse with the female gamete. And this natural population, it is also called as random mating population. Remember the other name, and also called as Mendelian population, the second name, and the third name panmetic population. And remember all these three names: the natural population, the Mendelian population, the random mating population, or the panmictic population. So what is panmictic? It was from the Greek Latin term where the pan means every, mixes means it is mixing. Okay, and why it is called as Mendelian population? Similarly, like we study in the law of segregation, where the alleles segregate independently, and in the law of independent assortment, as the character stays, there is no influence of one gene with the other. So similarly, like in this population, there is not at all any influence of any of the factors. They can freely intimate with each other, and every male individual have a chance to like fuse with the female. So which is a similar characteristics of Mendelian population. So that's why it is also called as Mendelian population, and it was for the first time used by the Wright. Okay, so this is what you have to remember regarding the population. This can directly come in the exam, the characteristics of the natural population. So what are the characteristics of this random mating population? See, whenever I say that some population is random mating, then it should follow some criteria. What is it? First thing is like there should be no control mating, and the pattern of transmission of the individual genes here in the random mating population cannot be determined, because we do not exactly know like which male is like fusing with which of the female within the population, because all are same, right? So individual transmission cannot be like tracked, and the genotype of the one or few individuals, it's not important, because here we are studying this as a whole as a population, and we cannot actually track like how the single individual is like fusing with the other female individual, like single individual is fusing with the other individual. And the gene pool and the gene frequency, which are the broader term, they are the one which are the most important in this population. We are studying the population as a whole, and we are considering mostly the gene pool and the allelic gene frequency or the allelic frequency into consideration. So what is gene pool? So gene pool is often confused with the other terms. I did get some doubt from the WhatsApp, not able to remember the other terms like which was mixed with the it is regarding genetic diversity, I guess. Someone was confused with these terms, genetic diversity and genetic stock. So all these terms are different, okay, they are not same. So specifically talking about gene pool, what is gene pool? Gene pool is a term which represents sum total of the actual definition, sum total of all genes that are present in the population of a species, specific species. Sum total of all the genes for all the traits that are present within the population of a single species, we call it as gene pool. And the change in, and there is a specific term called as microevolution. So what is this microevolution? Microevolution is like change in the allelic frequency within a single population over the time, and it is called as microevolution, which is observed for a short period of time. So within a short period of time, if there is a change in the allelic frequency within a single population, it is called as microevolution. And how this evolution is occurring? It is by the evolutionary forces which are mentioned here, which is mutation, migration, genetic drift, and the natural selection. See, these four parameters, these four factors are the most important for you people from the chapter of population genetics. So wherever you go, whatever you study, you always see these four terms. So please remember all these four terms. Do not ever forget it. So what is the meaning? Mutation is, no, you all know it. It is a sudden and irritable change. What is migration? Migration is nothing but to the set of population when there is a new individuals coming into that population, that is called as migration. And genetic drift, it is like a random loss of or random change in the allelic frequency, either due to the chance or by the sampling error. This is what the genetic drift means, and I have clearly explained, if you remember, about the genetic drift in my master classes. Okay, and next thing, these are the, again, these are the reasons of why the microevolution is taking place: the mutation, migration, genetic drift, and natural selection. I told what is genetic drift. Genetic drift is like change in the allelic frequency because of either sampling error or because of chance, but with no actual reason. That is what genetic drift means. And what is natural selection? Natural selection is a phenomena that was explained by the Charles Darwin, which says that in a population, some alleles will be favored, some phenotypes will be favored, and some phenotypes will be not favored. Whichever is favored or whichever has higher fitness will be selected naturally, and it will go to the next generation. That is what natural selection means. Okay, we'll discuss in bit detail. Now, next important thing before talking about the population genetics is like how to calculate the allelic frequency. Like remember, this is very important, like how to calculate the allelic frequency. Whenever I say allelic frequency, it is also called as what? Gene frequency. Do not get confused with this. So it can be represented as f of a, f of small a. Okay, so how to calculate it? So if I say there are 100 individuals, if there's I say there are 100 individuals and the number of individuals having the capital A is 80 and the number of individuals having small a is 20. So tell me, can you calculate the frequency of capital A and small a quickly? And if yes, like how do we calculate? Just tell me yes or no. Like, can we calculate the frequency of capital A and small a? Yes or no? Agar, so if you are feeling stressed, just watch this marathon series. Do not do anything at all. The stress will be relieved. Yes, we can calculate the allelic frequency. Okay, Musafir, do not worry. Like if you are stressed, like everyone will be stressed. If you ask anyone who is like preparing for the exam, even like see, I'm not writing the exam. Like I'm also already a PhD scholar, but still I'm tensed. If I am tensed, that then if you are tensed, it makes some sense. Okay, do not worry. That just accept it that this is like a situation where everyone will be tensed and even you will be, but you have to just face it. That's it. Face it with the confidence and just only focus with what you are revising and what you are doing and do it for next one another day. That's it. Don't waste your time on overthinking. So yes, we can do it. How we can do it? See, if the number of capital A is 18, number of small a is 20. If I have to calculate the frequency of capital A, it means what? Total number of A alleles in a population divided by total number of alleles. So that gives us the frequency of capital A. Oh, how much is the total number of A alleles? It is 80. What will be the total number of alleles? It is 2 into 100 because each individual, as I said, like it will have 2 alleles. Assuming that we are talking about a single gene with the 2 alleles, so then it will be 200, right? And how much it is? Like 8 by 20. You can solve it by yourself and you'll get some answer. And this will be the frequency of capital A. Similarly, like how to calculate the frequency of small a, it is like number of small a alleles now, it will be 20 and divided by total number of alleles into 100. You can do it into 100. So that will give you the frequency of the small a, fine. Or it is like just 20 by 200, okay. It is like 1 by 10 or it is 0.1, that's it. So this is the frequency, and if you want this frequency in the percentage, you can multiply it with 100. That's it. So that is how you can calculate the allelic frequency, just by having something in the given, something which is given. This is the prerequisite, and this can come in the exam where they will ask you to calculate the frequency of capital A, genotypic frequency of small a, or capital A, small a, or they can ask you the frequency of alleles like capital A, small a, frequency of capital A, or the frequency of small a. Okay, so how to calculate it? I told you this is how you can calculate it, and also this can be calculated by the number of alleles, or it can be directly calculated by the genotypic frequency. If, for example, they have given the genotypic frequency, capital A, frequency of capital A, frequency of small a, capital A, small a, and frequency of small a, small a. So if it is 0.36 and if it is 0.48 and if it is 0.8, so can you calculate the allelic frequency? Can you do it? If I have this, okay, good. So we can also calculate by using this genotypic frequency, where so the frequency of a allele will be number of individuals with a particular genotype with the total number of individuals in a population. Okay, that's how it is. Fine. Now allelic frequency, you have to know that. Let's talk about it later. But this point is important. The allelic frequency and the genotypic frequency, as I said, the value lies between the 0 to 1. Since we are talking about the frequency, it's like just the comparison between the different attributes. It will be like 0, like it will be, the value will be between 0 to 1. Okay. So next thing, now comes the actual principle of the Hardy-Weinberg law, which is the most important for your exam. Now I have given you the introduction about like what is population genetics, what is allelic frequency, what is genotypic frequency, what is population, and what are the characteristic features of the natural population, right? So now let's jump into the Hardy-Weinberg law, what exactly it is. See, the Hardy-Weinberg law is nothing complex. It is a simple formula or the principle which explains the relationship between the allelic frequency, that is like capital A, small a, with respect to the genotypic frequency, where if we have genotypic frequency, how we can directly calculate the allelic frequency, or if we have the allelic frequency, how we can directly calculate the genotypic frequency. That means like the relationship between the allelic frequency and the genotypic frequency over the generation to generation
is calculated easily by the equation that is given by the Hardy-Weinberg, which says that p square plus 2pq plus q square is equal to 1, where p plus q will be also equal to 1, and p square is nothing but like square root of the genotypic frequency, where p is like square root of the frequency of capital A, and q is the square root of frequency of small a, and we have 2pq, which is the frequency of capital A, small a, right? That's the whole thing here. Fine. This is what you have to remember, and this is the whole equation, which will be always equal to 1. So if we know the p square, we can directly reduce the p. So for example, if it is 36, if the p square is 36, can I directly tell that p is equal to 0.6? Yes or no? Tell me quickly. Tell me quickly, like if I know the frequency of the genotype, can I directly come into the conclusion that the value of p is 0.6? So now what is the value of p and q? P will be like the frequency of one allele, not always the dominant allele. It is not supposed to be the dominant allele always. It is just means that p is given to one of the allele and q is given to the another allele. That's it. If there is a trait with the two alleles, like gene with the two alleles, then it is prescribed for the one allele p and the q for the another allele. That's it within the population. And we come into the very famous, very important equation, which is like p square plus 2pq plus q square is equal to 1, where also p plus q is also equal to 1. But is it in all the condition that this works, that wherever I have a p square, I can directly deduce the p value? No, it has certain condition where this equation can be used. And Hardy-Weinberg principle, like what else is important for you in the exam? It is regarding like how this equation was like derived. So this is independently derived from both the scientists. So it is not the collaborative work. Remember, it is the independent work of both the scientists. And here, like about the Hardy-Weinberg, like also try to know about the complete name. It is G.H. Hardy. G.H. Hardy. He was from which place? You have to tell me. And this is like W. Weinberg, Wilhelm Weinberg, and he was from Germany. And Hardy from which place? Yes, quick. Yes, he was from England. See, also remember the places. Sometimes it might be tricky that they can ask you. For example, like the way you have in for the Mendelian genetics, like rediscovery, you have 3 different scientist names and where are they from. So those things are also important. Hardy from England and also Wilhelm Weinberg from the Germany. Okay. So what is the full form of GH? GH full form. Anyone? It is Godfrey Harold Hardy. I was not getting this term, like Godfrey. See, even I forgot this term, Godfrey. Remember Godfrey Harold Hardy. So you need not to like recall it, because in the, just like me, I have seen it here, like I got that this is the one right one. So while in the MCQs also, like if you do not know the exact and like name also, like if you have something somewhere in your mind, it will, you can easily able to understand. Answer it, do not worry. So firstly, it was a mathematical relationship between the allelic frequency and genotypic frequency, which was given by these 2 different scientists. And before that, the genetic equilibrium, which is the most important aspect of this principle, was proposed by the Yule, Cassel and Pearson at a different years. And later, it was by these 2 scientists who came up with this equation stating the law of genetic equilibrium under random mating. And it is also called as famously the Hardy-Weinberg law or the Hardy-Weinberg equilibrium. So what is it, equilibrium? The equilibrium in the sense, the gene and the genotypic frequency, like f of A, will not change from generation to generation. If it is generation 1, generation 2 and it generation 3, this will remain constant from generation to generation, but only if there is no external factors influencing this population. Okay, so what does the law states? The law states that gene and the genotypic frequency in a large Mendelian population or the large random mating population remains constant after generation after generation if there is no selection. This is important, please note, if there is no selection, no mutation, no migration or no random drift. or in a simple words, if there is no intervention from the any of the evolutionary forces, if there is no intervention from the human activities, the population will remain constant. The frequency of the different alleles within the population will not change. All the alleles in the population will be remain as it is, and they will pass on to the next generation. Okay, so this is what the entire understanding, like entire concept mean to say. And they famously derived a equation which is called as p square plus 2pq plus q square is equal to 1. Okay, so now what are the assumptions? So assumptions should be very important. You can directly get these points in the exam. First thing is population should be very much large. In a small population, there is like always a chance of genetic drift. So the population must be very large and the organisms are deployed, and this is only applicable where there is a sexual reproduction, where we have like male and female gametes, and the mating is completely random, and there is a normal meiosis and segregation, and there is equal fitness of all the genotypes. There should be like such that all the individuals in the population should be of equal fitness. There should not be any variable fitness in the population, and we are not talking about the overlapping generation. We are talking about the strict generation to generation. And the evolutionary forces like not acting, the one I said, like migration, selection, mutation, and the random drift. All of the these are the basic evolutionary forces that is responsible for the change in the trait, change in the allele, change in the genotype in the population, in any population you take. So none of this should act, only then this law will work. And what are the factors that influence? Same thing, the random mating will affect the Hardy-Weinberg principle, the natural selection will affect, migration will affect, and the random drift will affect. Okay, and the population size. So why it should be like population size should be large enough? So why it is so? Because in a smaller population, there is a significant genetic like random fluctuation in the allele frequency due to the genetic drift. So I'll tell you what exactly it is. See, consider two different things here. The baskets, like you have two different colors of balls. Okay, so first basket as like consider this as like 100 red balls and 100 white balls. Now just tell me like logical answer, like if I blindfold myself and try to pick up one ball from this, and no need to blindfold also, like I'll just try to pick up from one ball from this, or one ball is lost from here and one ball is lost from the second part. So we know in which of the following the complete variation is lost, the first one or second one, just by removing one ball, in the first case or the second case. A simple example, simple logical example why like genetic drift is have the more significant impact in the population. It's a second. See, just by removing only single ball, the only red is remain. If I remove the yellow ball, and there would be like no genetic variation at all, like only thing that is there is like red here. But if I remove like from the large set of balls, like if I remove the one ball of any color, still there is a significant variation where like you can still see like there are a lot of other balls with the two different colors. So this is how it is. By any chance, if one of the two alleles are lost when the population is so small, then the another allele which is present, it will be fixed. It will be fixed for the population, because there would be no variation. It will be there and it will be going on to the other generation, and the other one is completely eliminated. So that's the reason we always say that the genetic drift majorly affects, or it has a significant effect when the population size is very small. Okay, and you have to also understand that whatever explanation I have given regarding that there is no selection, no mutation, no random drift, this is not at all possible in the real population. Whatever real population we have in the nature, in none of the real population you see the ideal acceptance of this Hardy-Weinberg law. None of the population will actually follow it. All population will have certain degree of genetic drift. There will be like natural selection. All the evolution factors act on it. Okay, but this Hardy-Weinberg law can be like a control for understanding the law of nature, like law of evolution, like how it is going on. Okay, now what is the uses? Like uses, I came into the uses, what I was telling before. See, first thing is it is able to compute the genetic frequency from generation to generation, and for the evolutionary studies, like how exactly the evolution is happening, and it serves as a null model or the control model, and also it is used in the forensic analysis, and it acts as an experimental control. Okay, so these are some of the very important as applications of the genetic Hardy-Weinberg law, which you can remember as it is. No need to like go in depth about all of these things. Nothing much in detail will be asked. So just know about the its application. Majorly, it's about the allelic frequency and the genotypic frequency that you have to remember how to calculate it and how this Hardy-Weinberg principle is derived based on these two different factors, which is what allelic frequency and the genotypic frequency. That's it. You need not to worry about what is this forensic analysis, what is this null model, what is this experimental control. Forget about all this. Just know about this, the evolutionary studies. It is important because if we are able to know that like how the frequency is changing from year to year, we can predict something like, okay, whether the natural selection is acting or not, whether something random drift has occurred or not, whether there is a mutation in the population or not. All this can be predicted based on the values that we are getting for this allelic frequency and the genotypic frequency from the generation to generation. Okay. Next, the random drift, I already explained it, and this concept of random drift is given by the Sewald right. And I told you random drift is greater. Remember this point, this might be important, that random drift, the effect, it doesn't mean that random drift only takes place in the large small population. Yes, it takes place also in the large population, but its effect is greater in the small population compared to the larger one. Okay, it doesn't mean that random drift doesn't take place in the large population. It is like random. That's why the name random. Random change can take place in any of the population, but its effect is significant in case of the smaller population. I also told you like why it is so, right? So next, so these things are clear. You can skip it. Okay, and like relationship between the genetic drift and the population size. So I told you like the genetic effect of genetic drift is particularly high if the population size is low. Okay, so this is the exact relationship. And next thing, genetic drift and genetic diversity. See, if there is a genetic drift is occurring, see this ultimately leads to fixation of one of the two alleles. So this is its ultimate fate if the genetic drift is occurring over the years, over the generation, over the period of time. So ultimately, like one of the two alleles, if you are talking about two different alleles, one allele will be fixed and allele will be lost. So that's the reason the genetic diversity will decrease because of the genetic drift. Okay, this is its relationship. And what about genetic drift and heterozygosity? Same thing. Like since I told you like one allele is fixed, will there be like more heterozygous individual or more homozygous individual? Tell me. Like if the genetic drift is occurring in a nature in the population, whether there will be fix. If I say that there is a fixation of one allele, the percentage of or the frequency of heterozygous will increase or frequency of homozygous will increase. Yeah, I'll explain the bottleneck and Ponder effect. Don't worry. Yes, quick. More homozygous. So in the exams, if they are trying to ask you about the relationship between the genetic diversity, population size, and the heterozygosity, you should be able to clearly answer it. That even in the genetic drift, if it occurs, like it will heterozygosity will decrease from generation to generation. How much it will decrease? It is given by the heterozygosity in the next generation is equal to 1 minus 1 by 2n into number of generation into the initial heterozygosity. So this is the amount, this is the total entire value with how much it is decreasing year by year. Okay, this is what the heterozygosity equation looks like, and why it is 1 minus 1 by 2, everything you need not to worry it now. You know, if you know it, it's good, like I explained in the class, but if you do not know it, leave it. Just remember it is 1 minus 1 by 2n into number of generation into the initial heterozygosity. The final heterozygosity at some generation is equal to the sum value that is multiplied with the initial heterozygosity. That's it. Okay. Next thing, there are several ways how the drift can occur. I told you in the classes also, like there are several ways like how the drift can occur or how the population can change or the alleles can be fixed. Within that, there are two important phenomena that have been always explained. So one is called as bottleneck effect, other one is called as founder's effect. What exactly is this? See, bottleneck effect explains that this is the large population. For some reason, there is a drastic reduction in the population size. What are the some reasons I am talking about? Some reasons can be the natural calamities like floods or anything else like disasters. Because of that, the entire large population which has a lot of variation came into the small population. and this surviving population will be the one, the alleles which are randomly, which are by chance present in the surviving population, they will be fixed, and the final population will be having the reduced genetic variation. So whatever variation that was present in the original population, which is reduced now, and this reduced population, the alleles that are present, that will be fixed, and the final population will have the reduced genetic variation. This is how, like, one of the way of explaining the genetic drift, how it affects the population. Okay, and the next one, so the founder effects. Founder effects you can understand in this way: if this is a large population, over the million years, like if I say the population have been diverged, is it means that every individual in a population previously have undergone change? Is it means that if I say, like, is there is 100 years, from in the 100 years the one population is split into two, so that does it mean that all the individuals of the first population has undergone change? No, there will be a subset within the population, a small group of individuals which are actually the founders of the new species, or the new speciation, or the change. So that's what the founder effect means. Always in the within the large original population, there will be like very few who are called as founders that will form the separate new population, that is called as founder population, because of which there will be divergent and the variation splits. Okay, and the final population with which the new population is coming, obviously that will have a less genetic variation compared to its original population. That's what exactly the founders effect means. The new population which is formed, it is because of the founders, which are the small subset of population within the large population. Okay, large set of individuals. So this is about the two different phenomena which are actually related to the genetic drift only. Okay, remember that point. Next thing, numericals. I'll just try to solve one of the numericals. Please go through it and just tell me the answer. I'll allow you to solve. I will allow you people to solve this. You can solve it and tell me later. You can comment on this chat box. Anyhow, I told you, like, this PDF is available. In the PDF, I'll solve this answer and I'll send it to you. Okay, immediately after the completion of this live stream, not too much late. It won't be delayed. Okay, next thing. So this is all about the population genetics that you have to remember. You have to know about the Hardy-Weinberg law. You have to know about the allele frequency, the genotypic frequency, about all the evolutionary factors, which is like mutation, migration, selection, and which is the last point, mutation, migration, selection, and the random drift. Okay, and you have to know about the founders principle, like founders effect and the Bottleneck effect, and about the random drift, that it is majorly contributing, or the effect is majorly seen in the population size which is less, and what is the relationship between the random drift and the genetic diversity, what is the relationship between the random drift and heterozygosity, and what is the relationship between the population size and the genetic drift. So next thing, the next chapter which we are talking about will be like evolutionary genetics. So evolutionary genetics, let's talk about the different theories that have been put forward. The first thing is like theory of spontaneous generation. What does theory of spontaneous generation tells us? All the living organisms comes from the non-living organisms, which was originally proposed by the Aristotle, but this was like disproved by the Reddy and the Spallanzani, where they provided the experimental evidence, but finally it was resolved by the experiment conducted by the Pasteur and the Tyndall. That's what you have to remember from the theory of spontaneous generation. Next, with respect to the reproduction in the plants and animals, there are very important like events that have been taken place, where it was like these years might not be like important. You have to not remember all these years, but by any chance if they ask, there are possibilities. I can't say that there is a zero possibilities, but there are less possibilities, but the events are very important. De Graaf, he proved the tiny vesicles on the mammalian eggs were eggs, ovaries were eggs, and Spallanzani, he proved that the presence of sperm was essential for the development of progeny. And the conclusive proof regarding the fusion was given by whom? It was by Hardwick in case of C. urachin, and the Grieve, he described the stamens, and the Camerer told about the fertilization, where like seeds will not be formed without the pollination. And the another very important thing, it is about the first artificial hybrids in case of plants by the Thomas Fairchild, and it is famously called as Fairchild mule, and it is because of the crossing of Sweet William, which is Dianthus. Please remember the scientific name here, it is Dianthus barbatus into like Dianthus carophilus. Okay, This is famously called as Fairchild mule, which is like first artificial hybrid that was developed in case of plants, and you can at least remember this year. It is like 1717. Okay, and in the 1823, like Amici described the entry of pollen tube into the ovary. So these are some of the important events with respect to the reproduction in plants. Next, preformation. Before that, 1884, the fertilization event in the angiosperms was described by the Strasburger, and also the fusion between the egg and the sperm. Next, regarding the preformation. What is preformation? What does this theory? It was given by the Swaminathan and the Charles Bonnet. Okay, what is this actually? It says about what? The preformation, that there is some miniature body that is already there that will go to the next generation. That's what the preformation means. Preformation, it is already preformed, and this is again supported by the Aristotle, and this is the same concept what Aristotle explained. Based on this, there were two different categories of the supporters. One were called as animalculist, the other supporters were called as ovists. So what the animalculists believed, they believed that major contribution is from the uh, semen, egg, animalculist, and the semen produce only the stimulatory effect. They are called as animalculist, and they are called as ovist. And it was animalculist. They believed that major contribution is by the semen, and the nutrition was produced by the female. This is what they have believed, and they are categorized into two different categories. Okay, and next, according to both the groups, both believed that in the preformation there is a miniature body that is already prepared but not complete, but once the zygote is formed, this becomes complete, and that is how like new generation is coming forward. So this is what the preformation believe. Next, there is a epigenesis. What does this epigenesis talked about? It is by the Caspar Friedrich Wolff, who talked about the differentiation, how the organs are developed in the organisms. It is because of the process of differentiation. And later his famous successor von Baer told that the tissue development is through by the sequential modification of the embryonic tissue which are developed after the zygote is formed. Like because of the differentiation of the tissues, the new tissues are formed. So this is what the epigenesis explains us, which is like mostly accepted one of the organ development or the virgin of adult organs till today. Okay. Next, we have the one more important thing, which is like inheritance of acquired characters, which you might be mostly knowing, that was given by Lamarck. Okay, it's also called Lamarckism, and it was originally suggested by the Hippocrates. And the theory states that new character, it is acquired by the individual over its lifetime. It will be passed on to the next generation, and this new character, the change in the environmental factors, it will allow the body to accept or adapt to the new character. But this new character, it is also heritable. That's what is told by the inheritance of acquired characters. And this particular phenomena was also supported by the Charles Darwin. But is it true? Like the theory of inheritance, acquired inheritance, is it true? No. Okay. Just remember this. I told you about, if I have to recall what I told you about first thing, it was by the Aristotle, which we talked about theory of spontaneous generation, and next we talked about the reproduction in the plants, the different events that have been taken place, and next we came to the theory of acquired characters, and lastly, like we will be talking about the natural selection, which is the under the organic evolution, like how the evolution took place in the organisms was explained by the Charles Darwin with the concept of survival of fittest. And this is the phenomena where there is a phenomena where the natural selection occurs, where if there are all the population, all the individuals of the population will be divided into the fit and the unfit. Whichever is fit individual, that will pass on to the next generation by the selection of nature. The nature will actually select them, and they will be selectively promoted for the next generation, and which are weak, they will be selectively eliminated. So this is the whole concept of the natural selection. And during the development, this Charles Darwin, along with the organic evolution, he also gave one more theory, which is called as pangenesis theory. So what does this theory explains? It says that there is already a very identical small copy of itself of every organ is already prepared, which is called as pangene, where it will, after fertilization, it will go to its respective organ, and it will develop. And this was actually disproved by the Weizmann, who proposed the germplasm theory. So what according to the germplasm theory, Germplasm theory says that the body is composed of two different types of tissues. It is called as somatoplasm and the germplasm. Somatoplasm is like all body tissues which do not contribute for the sexual reproduction, and the germplasm produces the gametes for the sexual reproduction, and this is the only difference between the somatoplasm and the germplasm, where the gametes which are produced in sexual reproduction, these are only transmitted. The tissues or the traits that are formed in the somatoplasm, they are not transmitted to the next generation. Okay, and the fact that somatoplasm was not inherited was also showed by Castle and Philip. These are some of the very important theories that you have to remember from the theories of evolution. But yes, negated by, okay, the experiment of Weismann, good. So this is the some of the theories, but there are also some other things that we have to understand. So this is like regarding the origin of evolution and origin of species. I'll ask you one question. Tell me exactly like how exactly the life arose on the earth. So what's the one, can you just explain, tell me in the simple words or in the one line, how does the life arose in the on the earth? How? Simple question, that how life arose. I'm not talking about the human beings, life. I'm talking about the life. How did the life arise in the on the planet of Earth? I had my boost. It's now currently the secret of my energy. What do you think? Have you ever thought of it? Like, how did the life arise? Is it because like the one famous theory we know about, like inorganic to organic? Who proposed this? Inorganic to organic. All the organic molecules that are created, it was by the inorganic molecules. Anyone who proved this? There is also some experimental evidence for this. Yes, very good. So it is like Urey Miller experiment. But is it really true that the inorganic is actually leading to the generation of organic, where the life arises because of the non-living things? It's like similar to like what exactly is non-living to living, which was proposed by Aristotle. No, right? So still till today, if you want to know, like there is still today, there is no answer for this. That how did the life arise? Okay, there are different theories, there are different like speculations that have been made. There are different experiments that has proved few things, but not entirely. Like the things are very crystal clear about the evolutionary mechanism. This is how much it is untapped area. Like it is tapped, but like we did not get any proper conclusion for this. But whatever the current understanding we have to study it. So that's what I'm going to do it now. So entire evolution process, evolution is what, like evolution is change in the traits. Like we have to find out the answer why, how exactly the organisms are evolving, like what are the different mechanisms that is acting on the organisms in the nature. So all of this comes under the evolution. And and few things we also talk about the origin of species, like how exactly the life formed on the earth. So this is like there are a lot of theories, but they can be like, for the convenience of understanding, like they can be divided into four different types. Like that's what I always do, like I divide into four different types. What is it? Can you name them? First one is Lamarckism, second one is Darwinism, third concept is like mutation, fourth one, the latest one, the recent one, it is called as neo-Darwinism. Okay, so these are the major four like evolutionary theories which explains about the evolution and the origin of species. What is Lamarckism? Lamarckism, like theory of acquired characters, that we already discussed. It was by Jean-Baptiste Lamarck who gave this theory, which says that the living organisms and their organs tend to increase in the size during the evolution, and they are carried out to the next generation, which was not proved, right? Next, we have Darwinism. What it says about the theory of like origin of species. It is by the natural selection. So, but the problem with the Darwin theory is like Darwin did not explain about like how exactly the natural selection is acting. Now, tell me, if the population it is having 100 individuals, 100 individuals, all of them are same, all of them are exactly same. There is no difference in the fitness. Now, natural selection can act on this population. See, over the period of time, the population is like equilibrium. It doesn't change. The individuals are one and the same with each other. Can the natural selection act on this population, where it was explained by the Charles Darwin? Yes or no? Okay, talking about atomic fission. So they say there is no natural calamities that is occurring. There is nothing that externally the things are influencing. But is there any possibility that what is a prerequisite for the evolution then? What is a prerequisite minimum requirement for the evolution to take place? For any evolutionary method, like evolutionary factors to influence, what is a prerequisite within a population? Yes, diversity, and it's specifically called as variation. So variation within the population is mandatory in order to talk about any of the evolutionary process. So this was not explained by the Darwinism. Okay, not clearly. Like, as also he was believed that mutation is the reason, but it was not very clear. Okay, and he only emphasized on natural selection, and he studied, like, he went to two different islands, and there he studied like how the traits have been adopted based on like where they are living, based on the living habitat, like how the traits within the single species have diverged, and this was explained in the book of like with the book which was called as theory was fully elaborated in the Origin of Species, published in 1859. Okay, and next, yeah, this is all about the natural selection. So two main features, like if I have to talk about the features of like Darwin theory, like origin of hereditary variations in a population was explained, and there is selective multiplication of those variation, which we are talking about natural selection. Thus those variation will be like multiplied, which are the higher fitness. Based on the fitness, not only the fitness, like whichever traits, whichever individuals have the strength or ability to survive among the weakest ones, like compared to the weakest one, they will go to the next generation, the other one will eliminate. So that's what the theory suggests. Like if you compare the Lamarck theory and the Darwin theory, the difference is the change in the characteristics occurs by chance. This chance is actually the mutation we are talking about, and it is not influenced by the environment in which the organism is placed. It is purely because of chance. And the Lamarck theory suggests that organisms acquire the changes that enable them to adopt the environment, and these changes are inherited to the next generation. That was told by the Lamarck. And the mutations, here it was told that it is random, and according to the Lamarck theory, it is based on the adaptive or the directed mutations, not the random. Okay, so it says that like there is some reason like how the changes is occurring. It is based on like whether the organisms are utilizing it or whether the organism needs or not. That's based on that like the changes are occurring. That's what explained by the Lamarck theory. So this is the fundamental difference between these two theories. Next, we have like one more theory which is called as neutral theory of molecular evolution. Who gave it? It was by Japanese biologist, and it was by Moto Kimura. It was in 1960. He came up with the one more theory called as neutral theory of molecular evolution. See, all these things seems complex to you. I'll try to explain this in very simple words. I told you evolution can be in a simple words. It is like two-step process. What is a two-step? First thing is creation of genetic variation, that is like due to the mutation. So if the mutation is by the natural selection and the mutation continued by the random genetic drift, so this part is what the neutral theory suggests, and this part is what the Darwin theory suggests. That's it, very simple. You just understand this, nothing else, nothing more, nothing less. Just understand that the two-step process. First thing is mutation, common to both the theories. The next theory, it says that it is a random genetic drift, which is under the theory of like neutral theory of molecular evolution, and the Darwin more majorly focused on natural selection. He did not consider the genetic drift as a evolutionary force which is driving it towards the evolution of the organisms or the traits. That's it. Okay. Mutational theory. Next, we have another theory, mutational theory. This is regarding how the variations are created, and it was by Hugo de Virus who coined the term and also came up with this, observed this in the Ornithera Lamarckian. Okay, and according to the mutations, populations were pre-adopted, and adaptation did not rise because of the natural selection. This is what it believes. Now it is universally accepted that mutations are the one which are the ultimate source. Remember this point, ultimate source of all genetic variations, no matter like whatever evolution process that are present on the earth, right? But still the ultimate form of genetic variation, it is the mutation. Okay, Next, the last one, the neo-Darwinism. So what is neo-Darwinism? Neo is like new. Some concepts were added to the understanding of the Darwin and the complete theory was synthesized. But anyhow, like whatever might be, like we do not have the clear-cut answer for this. That's it. That's the ultimate thing. And the civil right, he proposed the shifting balance of the theory of evolution. What is this? You need not to worry. Just remember the name and the theory. That's it. The shifting balance theory of evolution was given by the civil right. So that's what important for you. So that's entirely completes your the different evolutionary forces, evolutionary theories that you have regarding the origin of species and also like how the evolution occurs. If I have to summarize, it started with what we talked about, the theory of inequity acquired characters. We talked about preformation, the pangenesis theory, the natural selection theory, and we talked about the 4 different categories of the evolutions study of evolutions, which is like Darwinism, neo-Darwinism, mutational theory, and the Lamarckism. That's it. Okay, and also one more theory which is like neutral theory on the molecular evolution. So if you have to know all the names of these theories and who gave it and what exactly these theories propose, that's it. This is what is important for you. Okay, next, mutation. Yeah, someone was talking mutation. So this is mutation. So just trying to correlate. I understand that you answered it for the previous question I asked. Now mutation. What is mutation? You know, it is a sudden but heritable change. Whatever the changes that occurs in the organism which is heritable, that comes under the mutation. Okay, and it is completely random. And the mutation. So what are mutants? Mutants are the one which is showing the individual showing the mutation. And what is variant? Variant is nothing but it is a altered phenotype. But until and unless it is understood as a mutation, it is a variant. If the variant is heritable, then it becomes the mutant. Okay, once it is like established that it is heritable, it is mutant, but before that it is just called as variant. And alleles of a gene producing the change in a phenotype, what we call, this is called as mutant allele. The other allele which is called as wild type allele or wild allele. Okay, and the mutations which are produced because of the change in the base sequence. If I talk about the gene, if this is a gene and these are the base sequences, and if the mutation is because of change in the base sequences, so this is called as gene mutation or the point mutation. And same thing, like the mutation is the ultimate source of all genetic variations occurring in any organism. So historical events, it was by Seth Wright. So first example or the first evidence of record of this mutation, it was discovered in a male lamb which was having the unusual short legs, which was dominant in nature. It was in Ancon breed of sheep. Remember this point, this is important. And the term mutation, I told you, it was coined by Hugo de Vares to describe the heritable change, but finally the systematic studies begin because of the white eye gene in the Drosophila by T.H. Morgan. Okay, you know it, like it was in 1910, which led to the discovery of so many other phenomena also. And next it was by H.J. Muller. So later it was also known that this mutation can be induced by the use of agents, where for the first time it was the X-rays which was used in the Drosophila, for which like he has also received the Nobel Prize. Later Stadler, what he did at the very same time, he also came to know about the effect of X-rays plus UV rays. Remember, this is very, very important. X-rays, you all know it, but if in exam by any chance if they are asking about the UV rays, also know he has studied both the X-rays and UV rays, the mutagenic effects in case of plants which are barley and maize. Okay, and the Orbach and Robson, what they have done, this is like they have studied the effect of the mustard gas in some other chemicals, and it was also in the Drosophila. They were the one who also told that other than the physical mutations, chemicals will also cause the mutation. This was done by the credit goes to the Orbach and Robson. Orbach is actually the female, remember. Okay, and mutational breeding, for the first time it started in Sweden, and in India it was in 1930s where the mutational breeding started in the plant breeding. Next one, characteristics of the mutation, it's the most important one. You have to remember all of these things. I'll discuss few things. What is mutable genes and what is mutator genes? So which undergo changes or which undergo mutation, they are called as mutable gene, and mutator genes which cause the other genes to undergo mutation, they are called as mutator genes. What are anti-mutator genes? Which suppress the mutation of other genes. So they are called as anti-mutator genes. And the mutation producing the same phenotypic effect may be located in the different sites. That means if it is a single gene, I am talking about A, if I talk about the base sequences, the mutagenic effect this site is producing we can have a same phenotype, and the mutagenic effect the other side is producing can also have the same phenotypic effect. So that's the nature of mutation. And it is also like forward and also reverse. Forward mutation is like conversion of wild type to mutant, and reverse mutation is like mutant to wild type. And if the mutants are not reverted any of the time, that then we can predict that the mutation is because of the deletion mutants, where the most of the part is deleted. Because see, if there is a single base pair change, this can be also reversed, where the same change can even occur. But even if certain region is completely deleted from the gene, so exact region getting reconstituted is near to impossible. That's why we say that whenever the mutants cannot revert back to the wild type, mostly like they are like deletion mutation. Okay, and the most of the mutant alleles produce a pleiotropic effect. What I mean to say is, if there is a gene and if there is some mutation that is acting on some base pairs or the chromosome region, so this will have not only have an effect on one phenotype, it will also have the effect on the several other phenotype. That's why the name pleiotropic effect. Okay, and mostly like frequency of the induction of mutation is higher at specific stage. Example is S phase, when where the replication is taking place. Okay, and the classification of the mutation is another important point, like where you have to remember all the terms here. What is forward mutation? I told you, like it is conversion from wild type to the mutant, that is called as forward mutation. Reverse mutation, it is like mutant to wild type. And the spontaneous mutations, which are naturally occurring, induced mutations, which are human made, and dominant mutations, where the mutant are dominant to the wild type. And the recessive mutations, the mutations are recessive to the wild type. Codominant mutations, like they are called codominant, partial or incomplete dominant, same just like what we have studied in the incomplete dominance, that whatever changes that we are seeing, it is like not completely dominant, it is partially. And somatic mutations, it is occurring in the axillary bud or the somatic tissues. Germinal mutations in the germ cells, and lethal, sublethal, subvital, vital and supervital, we talked about in the previous marathon series. Visible mutations, which we can see in the change in the morphology. And biochemical mutations, where there is a prevention of production of biomolecule, or any biomolecule like biochemical pathway is altered. So that's what the biochemical mutation is. And the macro mutations, which there is a major change in the phenotype. Micro mutations, there is small change. Null mutation and loss of function. So these two are often confused, the null mutation and the loss of function. See, the gain of function, what does it mean? Gain of function means like when there is a mutation, there is a gain of function. Some phenotype has some of extra effect. But what is null mutation and the loss of function? Null mutation is completely zero. Remember, it is completely zero. Whenever I talk about the loss of function, it is like decrease in the amount of phenotype that is provided. If it is like in case of enzymes, like decrease in the efficiency of the gene, which is having some of the role to play in some of the phenotype, so that will be like loss of function. But whenever I say the null mutation, the mutation is completely abolishing the role. So that's what the null mutation, and difference between the null and loss of mutation. Do not get confused with it, they are often confusing. Cytological basis, chromosomal mutations, detectable change in the chromosome structure. Gene or point mutation, as I told you, like it is a base pair alteration. And plasma genes, which is occurring in the plasma genes like mitochondrial DNA or the chloroplast DNA, that is called as plasma gene mutation. And next, based on the molecular basis, base substitution, where the nitrogen bases are substituted, that is called as base substitution. And deletion, where certain part of gene is deleted. And addition, where for the existing gene sequences something is added, like some sequences are added, so that becomes addition. And type of amino acid replacement, please remember, this is again very important point, and some things might confuse you. One thing is like missense mutation, where there will be a replacement of single amino acid with the another amino acids. That is what like missense mean. Replacement, the nonsense means like where the mutation is creating the nonsense codons. And the frame shift, where the entire shift of the reading frame, if it is AUG, JUG, and this is the 3D reading frame, this entire frame, if it is getting shift, that is called as frame shift. Neutral mutations, where you see the amino acid replacement, but there is no protein function that is changing. The protein function is same, the amino acid is getting replaced, but still the protein function is same. Lastly, the silent mutation. Silent mutation in the sense, you have the base substitution, the base is getting changed, but the amino acid is not getting changed. To which of the character of genetic code you can relate this last one, the silent mutation? Anyone? X-rays and Y-rays. What is this Y-rays? I did not understand, like, what is it Y-rays? Okay, I'll forget about it. Like, tell me, like, what is silent mutation? Like, it can be related to some characteristics of genetic code. What exactly it is? Where the change in the base is not changing the amino acid. It is still giving the same amino acid. Yes, very good. It is the behavior of degeneracy of genetic code. Okay, so this is under silent mutation. Okay, next, gene mutation. Gene mutation can be divided into two different types. One is spontaneous and one is induced. The spontaneous mutation, what is the rate of this mutation? 10 to the power of -4 to the -7. This is its rate. It's very, very rare. Okay, and it's 1 in 10 power 4 or 10 power 7. And this is about the spontaneous, and the mutations can be also induced, which are currently being done in the mutational breeding, where it can be divided into, like, spontaneous mutations can be divided into three different types. One is replication error, and second one is spontaneous lesions, and third one is transposition. Replication error, the mutation that is caused because of error in the replication. Spontaneous lesion, because of the DNA damage in the DNA. And the transposition, it is because of the presence of transposable elements. Okay, so this is about the three different types that you have to remember. One is because of replication error, second one is because of spontaneous lesions, and third one is because of transposition. Rest, like, you can study it on your own, like, just go through it once. And here, like, two things are important: transition and transversion. Transition is substitution from one pyramid into the another and one purine to another. This is transition. And transversion, what does it mean? It is like conversion of purine to pyrimidine or pyrimidine to purine. And number of total possible types, it is 4 in case of transition and 8 in case of transversion. Remember this is important. Okay, and I'll directly jump into the induced mutations because of the physical mutation. So till previous slide I talked about the natural mutations, but now we are talking about the induced mutations. Induced mutations, it can be because of so induced mutations can be because of physical agents, physical mutagens, chemical mutagens, or the biological mutagens. Biological mutagens are mostly the transposable elements. Okay, the chemical and physical we'll talk about. The first one, the physical mutations, they can be divided into two different types: ionizing and non-ionizing. And the non-ionizing, the only one example we have, it is UV rays, and which will lead into the formation of thymidine dimers. Okay, this is the one example. And the ionizing radiations, we have again two different types: particulate and non-particulate. And the example for the particulate, alpha rays, beta rays, the fast neutrons, and non-particulate we have gamma rays and the X-rays. So these are the examples. Okay, that's what you have to remember. So the complete story is done. The physical mutations, ionizing and non-ionizing. And in the non-ionizing, only one example is UV rays. For the ionizing, we have two different types: one is particulate, one is non-particulate. And here, in case of particulate and non-particulate, remember, like, ionization, order of ionization: alpha, beta, and gamma and X-rays. Okay, and regarding the penetrating power, the gamma and the gamma is highest, next X-rays, rest all of this alpha, beta, and fast neutrons. Okay, and here in between this, the fast neutrons comes. So this is about the ionization power, the order of ionization power, and this is about the penetrating power. It is like just opposite to each other. The penetrating power will be higher to the gamma rays, where exactly opposite the ionizing power will be highest, densely ionizing. We call it as densely ionizing term. This is highest in case of this ionizing radiation, which is like alpha, neutron, and beta, then the gamma and the X-rays. Okay. One thing, like, okay, I'll talk about it. Next, I'll talk about the chemical mutagens. See, chemical mutagens are the one where the students are mostly confused about. See, in case of chemical mutation, let's try to simplify it. See, they are like different categories. One is base analogs, second one is base modifiers, and third one is intercalating agents. So what is intercalating agents? What is, like, I'll discuss all of these three. Base analogs, as a term says, there are some chemicals which are completely analogous to the existing base, like 4 nitrogen bases, that is like structurally, functionally same. So that is what the base analogs means. Base modifiers, there are some chemicals which will modify the bases. This can be again divided into two different types. One is deaminating agents, and the other one is alkylating agents. And the last one, intercalating agents. That means they can come in between the two nitrogen bases and place themselves. That is what the intercalating agent means. So for you, for the exam, remembering the example is important. The base analogs have the strong structural resemblance with the normal nitrogen base, where it can be incorrectly added. So that is 5-bromouracil, chlorouracil, iodouracil, aminopurine. All of these are the examples. Remember the examples for the base analogs. Similarly, for the deaminating agents, this three important is the most important one, where because of the deamination of cytosine, if the cytosine undergoes deamination, then it will form actually uracil. So initially the cytosine has a base pairing property with guanine, but the uracil, it has a base pairing properties with the iodine. So there is a shift, there is a complete shift in the base pairing because of the deamination. Next, the deamination of adenine will gives you the hypoxanthine, which has a base pairing properties with the cytosine. Originally, A will pair with T or U, but the hypoxanthine will pair with C. Okay. Next, the deamination of guanine gives xanthine, but this does not have any mutation effect because the guanine base pairs with C and xanthine will also pair with C. Okay, but these three are important. It will be directly asked in the exam. Please remember the deamination here of all the nitrogen bases. Next, we have alkylating agents. The examples are ethyl methyl sulfonate, EMS, MMS, methyl methyl sulfonate, nitrosoguanidine, and dimethyl nitrosamine. All these are some of the basic examples, very representative examples for the different alkylating agents. What are these alkylating agents? It is like adding of alkyl group. The base will be added with the alkyl group, which will change its base pairing properties. So how exactly? See, alkylation of guanine. So what happens? Like it will give 6-ethyl guanine, where it will pair with the thymine. Originally, the G has to pair with C, but after conversion of alkylation, it is pairing with T. So you are seeing that from GC there is a AT transition, one purine to another purine. Next, EMS, the ethyl thymine, it is converting into ethyl thymine. The originally thymine will base pair with what? A, but it is now base pairing with G. So where there is a transition from the A to G, transition from A to G, again from A to G, purine to purine. And the third one, cytosine, it is hydroxylates NH2 of cytosine. Because of this hydroxylamine, so this is giving or pairing with the adenine. Originally, so the cytosine will pair with G, but now this is pairing with the adenine. Okay, so this is what you have to remember. The examples are very important: EMS, MMS, and this conversion and the above deamination reaction. All this will be the one which you have to remember for your exam. Focus on these things, revise the terms properly so that you won't be confused or you won't be like forgetting this. Okay, intercalating agents. I told you about this intercalating agents and the detection. See, this completes about the mutagens. I quickly have completed it. I did not go in detail anything. So that's not required for right now. What is important for your exams to know about the physical mutagens: ionizing, non-ionizing. In case of non-ionizing, it is EV rays. Ionizing, it is particulate, non-particulate, which is densely ionizing, which is sparingly ionizing, which is high penetrating power, which has low penetrating power. Next, talking about the chemical mutagens, they can be divided into base analogs, base modifiers, intercalating agents, and again base modifiers can be divided into deaminating agents, and it can be the alkylating agents. And deaminating agents, there is a certain change in the base pairing properties because of deamination of certain nitrogen bases, which you have to remember. And also in case of alkylating agents, because of the alkylation, adding of the alkyl group, the base pairing properties changes, where we can see the effect of transition. Okay, intercalating, I did not talk about it. So where the insertion have been done between the two adjacent base pair. If it is there is a two adjacent base pair in the gene, so there is another chemical which is being added in between. Okay, and they are considered as a powerful mutations in case of prokaryotes. And the examples are propylene, aquadine orange, and yttrium bromide, ICAR compounds, and acrophamin. All these are the examples for the intercalating agents. Okay, next. In case of detection of mutation also, like you need not to refer anything in detail. I'll tell you the one simple thing here. In case of prokaryotes, you have two different tests. One is replica plating technique. and other one is like Amis test. Who gave this? Can you tell me? This is by Bruce Amis, and the replica plating technique, Esther Liederberg and the Josa Liederberg. Remember this. Okay, and this is in case of prokaryotes. In case of eukaryotes, we have CLB stock, attached X chromosome, and the Russell's dominant lethal test. It is specifically in case of maize, and it was given by LB Morgan, and CLB technique, it was given by Muller, and Russell's dominant test, Russell in case of mice. So this completes your detection of mutation. That's it, nothing else. In the prokaryotes, you have 2 different tests to remember. In the eukaryotes, you have 3 different tests to remember. That's it. Clear to everyone? Is it done, mutation part? Susan, is it okay? Satisfied? It's not about oval hypothesis. Degenerate is right, as I told you. Okay, fine. So this is all about the mutation. This is all about like the population genetics I wanted to discuss. So I asked you one question that I will post it in the pin it in the this one comments also. You can comment your answer on that. Okay. So thank you so much. Like the other thing which is left is history and cytogenetics part, the chromosomal aberrations. So I'll try to get it done by before evening. Should I do it or like it's fine? Like if you are in, like if you will be able to attend it or if you will see it later, I'll do it. Not a problem for you, for me. Tell me like if I have to do it, even if like one student is expecting it, I'll do it. The history part and the cytogenetics part. Okay, so if you want it, I'll do it. Thank you so much. Like you have one day of time. Do not worry. Like do not worry. Be calm. Like whatever you can revise it till the evening. If you want to sleep for some time, have a good nap in the afternoon and wake up in the evening. We'll again meet in the evening session and make sure to plan such a way that you sleep early in the night today. Okay, at least by 10 p.m. or maximum 11 by 11 p.m. Since you have to wake up minimum of by 5 or 6 in the morning. Centers might be far away, or even if it is near also, like anyhow you have to reach by 8:30 or before 9, as I told you in the video also. So please try to make a schedule where you sleep early for today. Rest we can discuss in the evening ones. So all the very best. Prepare well. Do not panic. Thank you so much for attending. Yeah, I'm uploading this file. It will be uploaded in like another 10 minutes. Okay.